The fasting cycle is really just as important, and perhaps in some ways more important, because things are less available. Nutrients are less available. So the body has to be a lot more creative in what it does for its metabolic needs. Okay? So the fasting cycle is referred to also as post-absorptive. Okay? Again, terminology is very important. Post-absorptive is synonymous to fasting. Obviously, the main thing is you're not going to have glucose. Glucose is dropping. Tags are dropping. Amino acids are dropping. There's less of all three in the bloodstream. So what does the body have to do to compensate for the lack of nutrients? It will go to what is stored. What is stored? Glycogen. It breaks down glycogen to provide glucose. What else is stored? Tags. It'll break down tags, provide fatty acids as an alternative fuel. It will turn over, break down some protein to get amino acids so you can conduct gluconeogenesis using the carbon skeletons of these amino acids. So you are primarily in a catabolic phase where you're breaking large molecules into smaller molecules that the body needs to operate. Okay? So we'll cover some of the key effects on each of the main organs we've been talking about, and we start with the liver. So the liver is the main, although you have glycogen in muscle, the important glycogen storage resides in the liver, because only the liver can produce glucose. The muscle cannot. The muscle produces glucose 1-phosphate, then glucose 6-phosphate. That does not leave the muscle, is unable to leave the muscle. So only liver glycogen is relevant. So glycogenolysis will be increased with the purpose of delivering glucose to the bloodstream so the organs that need it can use it. The liver is the only organ with, the other organ that can do that to a smaller extent is the kidney, that can do gluconeogenesis or has gluconeogenesis. So there's increased gluconeogenesis. So what you think here for in terms of fasting, you are providing glucose to the bloodstream to maintain it at the normal levels. Okay? So the liver's function in fasting is becoming a factory of glucose. It's a glucose producing organ. It's switched from a glucose consuming organ in the well-fed state to a glucose producing organ in the fasting state. In many ways, the liver is kind of altruistic, meaning that it cares more about the rest of the organs than about itself. It is providing glucose for the use of the brain, red blood cells, and other tissues that are entirely dependent on glucose. The liver itself will switch for its energy needs to beta oxidation of fatty acids. It's no longer using glucose in glycolysis. It's using fatty acid beta oxidation for its energy needs. You produce a lot of acetyl-CoA. Acetyl-CoA goes to the TCA cycle, produces more energy for the liver. At some point, the liver has enough energy, then acetyl-CoA starts to accumulate. And then that acetyl-CoA is used to make ketone bodies. So the liver then is providing another source besides the glucose, is providing another energy molecules in the form of ketone bodies. Again, the liver itself cannot use ketone bodies, but it's providing ketone bodies so other tissues that can use it will pick it up. And many tissues can use ketone bodies. Skeletal muscle, heart, even the brain can use ketone bodies. That's the only other form besides glucose that the brain can use. Okay? Adipose tissue, key thing in adipose tissue, you always want to think of it as it is our main store of tags, of fuel, lipid fuel in the body. Don't think of adipose tissue as a bad thing. It's really a good thing. We depend on adipose tissue. We don't want too much of it, but we depend on it for our energy needs. When we are fasting, when we are sleeping, when we haven't had a meal for several hours. And just kind of while I am thinking about it, fasting is typically 4 to 5 hours after a meal and beyond. Okay? After 4 or 5 hours from the time you've had your last meal, that's considered fasting. What's also considered fasting is obviously sleeping. If you're sleeping, you're fasting. So you're going to need glucose even when you sleep. So you're going to have glycogenolysis and gluconeogenesis during your sleep. Okay, back to adipose tissue. The adipose tissue is the major depot, if you want, of tags. During fasting, you want to think of two hormones that are going to be elevated in the bloodstream, glucagon and epinephrine. And both of them are going to result in, or going to control much of metabolism. Epinephrine in particular will activate the beta-adrenergic receptor, activating cyclic AMP, activating PKA. PKA phosphorylates hormone-sensitive lipase, which breaks down tags in adipose tissue, producing 3 fatty acids and glycerol. All 4 molecules are released in the bloodstream, transported in the bloodstream. Fatty acids usually are bound to serum albumin, and they are picked up by various tissues that can use them. Muscle, liver, heart can use fatty acids as well. So they're picked up to be used in beta oxidation. To the muscle, remember here, you have no insulin. This is a kind of key point. You have no insulin when you're fasting. You have glucagon and or epinephrine. So the muscle uptake of glucose decreases. It's no longer picking up glucose because of the absence of insulin. Instead, the muscle will start to pick up fatty acids from circulation, use those fatty acids for its own energy needs. Okay? It can use ketone bodies as well, produced by the liver. So it switches to fatty acids and ketone bodies for its own energy. The muscle has another important role in fasting, in that it can provide amino acids. The muscle is rich in protein. Muscle proteins, structural, functional proteins that turn over normally. Muscle proteins typically have a high turnover rate, meaning they're degraded usually pretty frequently and replaced, degraded and replaced. When you degrade those proteins, you produce amino acids. Those amino acids are released in the bloodstream, transported to the liver, where they can be used in gluconeogenesis. Okay? The brain is really kind of dependent on the liver for its glucose, because the liver is producing the glucose, as well as ketone bodies. Both of them are produced by the liver in fasting. However, ketone bodies in the brain are only picked up after a prolonged fast, not in the first, say, few hours or days. It may take several days, sometimes a week or two, before the ketone body levels reach the concentration that the brain needs to pick them up. Okay? But nevertheless, the brain is able to use ketone bodies. The brain cannot use fatty acids at all because of the blood-brain barrier. Fatty acids will not, most fatty acids will not cross the blood-brain barrier, whereas ketone bodies can and glucose can. Okay, I'm taking a little detour here because this is relevant in the fasting state. We were talking about that you enter a catabolic phase when you are in the fasting state. And that means you are going to degrade glycogen to form glucose, tags to form fatty acids, and protein to get amino acids, and so on. Well, I want to just show you how the reserves of the body, let's say we're in a fasting state, this is kind of the relative amounts of reserves that are present in the body. I want you to start with here. So this is a 70 kilogram person. Of their weight, 0.2 kilograms is in glycogen. That's a very small amount. That translates to about 800 calories. There's a lot more proteins in the body, and that's equivalent of this leg, about 5 kilograms, 6 kilograms. And that translates, when you degrade that protein and the amino acids, into about 24,000 kilocalories. But look here. That's the amount of fat reserves we have. 15 kilograms. That's nearly 20%, actually more than 20% of body mass is in the form of tags, essentially. This is tags. And look at the number of calories it can provide. 135,000, 5 times more than protein, and more like 140 times what the glycogen can provide in terms of energy. So these fuel reserves are really important. To be aware of them is really important. Okay? Now I'm going to go through here and just remind you of the two important concepts here. First of all, glycogen gives you glucose directly through glycogenolysis. Proteins, most proteins will be degraded to amino acids. Let's say you get alanine here. Alanine can be converted to pyruvate. Pyruvate can be converted to glucose. Okay? So most amino acids can be converted to glucose as well to give you energy in glycolysis. However, fatty acids have a different fate completely. Fatty acids, first of all, this is tags stored in adipocytes. They're going to produce fatty acids. Fatty acids go through beta oxidation. By going through beta oxidation, they produce energy directly. They produce acetyl-CoA that goes through the TCA cycle, produces more energy. So fatty acids will produce, beta oxidation will produce energy in the form of NADH, FADH2, and some GTP and so on. And all of them will be converted to ATP. So fatty acids, beta oxidation produce energy directly. Note that fatty acids cannot be, beta oxidation does not allow acetyl-CoA. What you produce from beta oxidation is acetyl-CoA. Acetyl-CoA cannot be converted to glucose. That's a very important concept. So fatty acids can provide you energy, but they cannot be used to make glucose. Glucose can come either from gluconeogenesis or glycogenolysis. Okay.